Fiber photometry calcium imaging combines genetically encoded calcium indicator and multimode optical fibers to monitor neural activity in 3D moving animal, which is critical to understand how a specific group of neurons play in directing or responding to an action or a stimulus. This technique is an accessible way to record from specific brain regions that are defined by their connection or genetic profiles, with the built-in control to separate signal to noise. Demonstrating the procedure will be Ekaterina Martianova, a graduate student in my lab.
Begin by loosening all screws on the 5-axis translator. Screw in the patch cord to the adapter that is affixed to the 5-axis translator. Then, turn on the 470nm excitation light at low power and position the tip of the fiber pointing to an autofluorescent plastic slide. Next, record from the complementary metal oxide semiconductor or CMOS camera in live mode. Increase the gain or adjust the lookup table until the image is visible at the focal point of the objective. Advance the 5-axis translator towards the objective, ensuring that the 470nm light is centered on the fiber at the SMA or FC end of the patch cord until an image can be resolved on the camera. Finally, adjust the X and Y axes until the image is centered and well resolved. Visualize the light emitted from the ferrule end of the patch cord as it should appear as an isotropic circle.